All right, good morning, everybody. Today we're going to work on some genetic technologies that we have. And if you want to take notes while we're doing this, I would kind of suggest you pause it and set it up like this. We're going to talk about plasmids, transgenic organisms, cloning, and gel electrophoresis. Okay? All right, so genetic engineering is actually a broad term of what we can do with some parts of cells and DNA and different things. So, engineers create new things, and that's what genetic engineering is. So, the first example is what we can do with a plasmid. A plasmid is a ring of DNA that's in bacteria, and we can actually insert genes into that plasmid to get the bacteria to make things they normally didn't. So it's called recombinant DNA because we've combined different pieces of DNA. So what they do is with chemicals, they cut the ring, they put the gene that they want in there, and then as the bacteria reproduces, it will now make whatever gene that we've inserted. The best example is our modern insulin gene. Okay, so here's a picture of that. Here's the gene we took out of our human cell. Here's the ring of bacteria from the ring of DNA from the bacteria. And then we cut that and splice it in, just like they used to do with old tapes and stuff. Then they reinsert it back into the bacteria, and now the bacteria will make a hormone or something that it didn't before. In this example, it's human growth hormone, but it can also be insulin. Okay, so if you're writing stuff down, these would be the 3 points that I would make. It's a ring of DNA in the bacteria. We can insert the desired gene that we want. And then the best example is insulin. Okay, so the next one, transgenic organisms. These are just organisms that have foreign DNA in them. So we just talked about the bacteria that we can manipulate. We also have transgenic plants. We can reduce the amount of frost damage. We can naturally reduce insecticides so the bugs don't eat them, but we don't have to spray all these chemicals on them, all kinds of things. And then we can also produce animals that might grow faster or larger. So I want you to think about the advantage of doing these things would be for ethical reasons, right? Like feeding more people or having multiple plants grow in one season, medicines, different things like that. Okay? All right, so here are some examples of those, and we won't talk about all those. We won't talk about these necessarily, but these are making plants grow faster. These are making fish grow faster and larger. They've inserted the jellyfish gene into these pigs so they can glow, which there's no real ethical advantage to that. I think they're just seeing if they can do it. And they actually had a mouse grow a human ear on it one time to serve as a prosthetic. That was many years ago. Okay, all right, so if you're writing things down, transgenic organisms, any organism that has foreign DNA in it, and I want you to think of it, any reason to make society better. Food, medicines, something like that, and we can do it for bacteria, plants, or animals. Okay, all right, the next one is cloning, and this is obviously making an identical organism from donor cells. I do want you to see how complicated it is. It's not like the movie The Fly where you just push some button and there it goes. So Dolly was the first mammal to be successfully cloned, but what they didn't report in the news is that it actually took 101 tries for this to work. So here is the sheep they cloned. They took a cell from her udder. Okay, here it is, and they took an egg cell from a separate sheep and they removed the nucleus from that. So see how it's basically like an empty shell, so to speak. And then they electrically fused these two together, so then the nucleus from this sheep became housed in the cell from this sheep. Okay, so here's that cell, and then it's kind of like what they do with in vitro fertilization and stuff. They put it in a petri dish and it started dividing on its own. And then once the embryo reached a certain size, they actually surgically implanted into a third sheep who was at a particular point in her cycle. So it's almost like she became the foster mom to this developing baby. Okay, then she actually gave birth to Dolly. And Dolly is an exact clone of this particular sheep here. Okay, so that's how cloning works. It's long and difficult and it has a lot of ethical issues along with it. Okay, so this is what I would write for that. Identical organisms, long, difficult process with lots of ethical issues. Okay, all right, and then the one that is actually used the most for crime scenes and paternity identification and those kind of things is DNA fingerprinting. So keep in mind this has nothing to do with your actual fingerprints, okay? The reason it's called fingerprinting is because it's unique to you, just like your fingerprints are. So we use it to solve crimes, find paternity, study genetic disorders. They even use this to see if organisms are more closely related to another. Okay, so here's the process for this. Just think about a long DNA strand. We have chemicals called restriction enzymes that look for certain patterns in the DNA, and they cut them. So basically, if you can think of your DNA strand like cut into thousands and thousands of pieces, and then if we can compare those pieces from organism to organism, you can kind of get some similarities in there. Okay, so once you cut it up and you put it in these things called wells up here, you run some electricity through it, and then you end up with these bands. Okay, DNA is negative and it's attracted to positive. So if you notice the anode and cathode here, small pieces move really quickly, larger pieces move slowly. Think about if you're at a stoplight and you're in a Mustang or some other little sports car and there's an 18-wheel truck next to you. You are obviously going to take off a lot faster than he will. Okay, so you're going to get further down the gel, in other words. All right, so if you're going to write something down for this, running electricity through DNA that's been cut, it helps you identify paternity, criminals, common ancestors, things like that. Okay, so guys, here's an example of a DNA fingerprint, and these are actually much more complicated than you'll see, but see how you can match them line for line? So here's your evidence, which I don't like that it's in the middle, but see how sample 2 matches line for line for line. Okay, so that tells me that sample 2 has something to do with this crime, if that happened to be a crime. Okay, and then over here, you can see the 2 samples that match are 1 and 4. Okay, so that's one of the ways they can use that. All right, so for this example here, suspect 3 is the one who was accused. Do they have the right criminal? Well, if you look at row 3 compared to the evidence column, you'll see immediately with the first and second bands that they don't match. Not only do they have to be in the same location, they have to be the same thickness and everything. Okay, so suspect 3 is not the criminal, or at least that's not the man or woman we have evidence for. Okay, so who do you think we should actually question? I would think it would be person 5. See, the thickness matches, the placement of the lines match, and everything. Okay? All right, so same thing here. Here, V is victim, E is evidence, S is your suspect. The evidence does not match the suspect in the top answer, but in the bottom one, it absolutely does. Okay? So when you're doing criminals, the lines have to match every single one, 1 for 1. All right. Now, when you're doing paternity test, it's actually a little bit more complicated because if you remember, every child receives 50% of the DNA from the mom and 50% of the DNA from the dad. So half of the child's lines should match the mother and half should match the father. So if you'll notice, we have mother, child, father. So in this first instance, they matched the kid to the mother first. And guys, remember, most of the time the mother's not the one in question, but we have to know which genes to test against the father. Okay, so in this case, see how the ones that didn't match the mom match the dad. So now we know we have this father. Okay, in the bottom example, however, the ones that didn't match the mom don't match this dad. Okay, so they cannot prove that this particular guy is this child's father. And that's how they do that on Maury or any of those other talk shows where you've seen paternity testing. Okay? All right, so I'm going to give you guys some things to practice to try and do some of these things. Just remember, if you're matching a criminal, every line has to match. And if you are matching a baby, half have to match the mom and half have to match the dad. Okay? Thank you. Make sure you email me if you have any questions or concerns.